Hello everyone. In this video we will discuss the question number 36 of GATE 2019. Which one of the following covalent linkages exists between 7-methyl guanosine, M7G and mRNA? So this question came from the chapter of mRNA processing. So as you know that in the 5' end of the mRNA, that 5' end will be capped by a guanosine, which can be modified by methylation at the N7 position. So that is 7-methyl guanosine, which is a which is a G cap at the 5' end. And so they are asking that which covalent bond exists between this G cap 7-methyl guanosine and the mRNA transcript. So the options are 2' to 3' triphosphate, 3 to 5 triphosphate, 5 to 5 triphosphate, 2' to 5' triphosphate. So let's first briefly understand that what are the events which are occurring between the mRNA 5' capping of the mRNA. So suppose this is a 5' end of the mRNA and this is a growing mRNA which is being transcribed from the coding DNA. So this is the nucleotide which is at the 5' end, right? N means any nucleotide it can be, and it will have 3 phosphatases at alpha, beta and gamma position. So the first step, the phosphohydrolase will come and it will remove the gamma phosphate. So now this 5' end nucleotide will be having only alpha and beta phosphate. After that GTP will come, and GTP it has guanosine along with the 3 phosphate at alpha, beta and gamma position. So this beta gamma diphosphate, it will be released as a form of PPI, right, or pyrophosphate. So GTP will be so after this step GTP will be remaining with only one phosphate, which is alpha phosphate. So GTP will be converted to GMP where phosphate is in the alpha position. So now this GMP with alpha position phosphate, it will make a covalent bond with this 5' nucleotide of the mRNA, right? So this will form this complex where this is your 5' end and this is the G cap. So these two phosphates are from the alpha and beta phosphate of the 5' nucleotide of the mRNA, and this is the GMP where the phosphate is at the alpha position. After this there can be methylation at the G cap. So for the methylation it will use the adomate as a cofactor. So as you know that met stands for methionine, right? And methionine has a methyl group. So this methyl group of a methionine will be transferred to the N7 position of the guanosine here, right? So methyl group of methionine will be transferred to the N7 position of the guanosine, which is the G cap, and it will make 7-methyl guanosine. So and so it will form this complex, right, where guanosine is methylated at a position N7 position. So now if we will see the structure of this complex, or then we can see that this is your mRNA transcript, right, and this is the 5' nucleotide, right, nucleotide at the 5' end of the mRNA transcript. So it has alpha phosphate and beta phosphate, right? Here also it will have alpha phosphate and beta phosphate. So this 5' nucleotide of the RNA has one alpha phosphate, another one is beta phosphate, and it will make a covalent bond with the guanosine, right? Guanosine, which is methylated at the N7 position to produce 7-methyl guanosine. So this 7-methyl guanosine will make a covalent bond with this 5' end of the mRNA. And how? And also the 7-methyl guanosine has a phosphate at the alpha position, right? This one. So 7-methyl guanosine has a phosphate at the alpha position, and this 5' nucleotide of the mRNA, it has 2 phosphates at the alpha position and beta position, and they will make a phosphodiester bond, right? Phosphodiester bond they will make. This is the part from mRNA, right, and this is the part from the alpha phosphate of GMP, which is a G cap, or actually modified GMP, which is 7-methyl guanosine. So this phosphate is the part of the G cap, and this two phosphate alpha and beta. This is the part of your 5 prime end of 5 prime nucleotide of the mRNA. So this is the part of the mRNA and this is the part of the 7-methyl guanosine G cap. So they will make a phosphodiester bond, and this phosphodiester bond is known as 5 prime to 5 prime triphosphate linkage, because the phosphodiester bond has been formed between this beta phosphate of mRNA and alpha phosphate of 7-methyl guanosine, right. So so this beta phosphate is at the 5 prime position of this 5 prime nucleotide, right. This both of this alpha and beta phosphate are at the 5 prime position of the beta phosphate of the 5 so these two phosphates, alpha phosphate and beta phosphate, they are at the 5 prime position of this mRNA nucleotide, right. And this alpha phosphate, which is a part of the G cap, that is also at the 5 prime position, right. This is the, so this alpha phosphate of GMP is also a part of 5 prime position, and this alpha and beta phosphate which belongs from the mRNA, that also resides in the 5 prime position. So that's why this linkage is known as 5 prime to 5 prime linkage, right. And there is involvement of 3 phosphate group, right. So that's why alpha, beta and alpha. So that's why this phosphodiester bond is known as 5 prime to 5 prime triphosphate linkage.